Welcome everyone. Thank you for coming to this student seminar. Today we have my PhD student, Ayesha Vermani. She's in the program in neuroscience. She's been working with me for a while now. Important thing is that she has a cat. I don't know if the cat is with her right now. Usually in our one-to-one meetings, the cats, show up on the screen. It really makes me happy. Anyways, I think she's going to show some cats, but the talk is not about the cat. It's a neuroscience talk, and she's going to tell us about the dynamics and codes of neural activity and how we might analyze it better, and some modeling efforts in that direction. All right, Ayesha, take it away.
You're muted.
Oops. Yes, my bad. Okay, so hi everyone, I'm Ayesha, and today I'm gonna try to answer this question, can time-dependent and invariant decoders coexist? But before I go into the details of this, let's ask a more basic question, which is, how does the brain represent information? And let's assume in this case that the cat is the brain and it sees a squirrel. And in response to the squirrel, the neurons in the brain of the cat respond by firing action potentials, which I represent with these ticks are. And in fact, it's not just a single neuron, but a population of neurons respond by firing over time to different features of the squirrel. And let's say the squirrel also sees the cat and tries to make an escape by hiding behind a tree. Now the cat has to maintain the position of the squirrel along with different features of the squirrel in order to make its next decision. Now, when we study neuroscience in a lab setting, we typically train animals on behavioral tasks in a more controlled way, while we record from different areas of the brain. For instance, I'm going to talk about this one task here, where the authors train monkeys to discriminate between two tactile frequencies. So they were presented with one frequency, and they had to maintain that frequency in their memory for several seconds, so they could compare it in order to do the task well. And while they were performing this task, the authors recorded from the prefrontal cortex, which is roughly in this region, and it's been implicated in various functions, including short-term working memory. And then they looked at how the neurons that they recorded from respond during this delay period. So how they're plotting the activity of 3 neurons. And it's conditioned on different frequencies, and this is just the average. So they do notice that there are some neurons that fire persistently, that show differential firing persistently for different stimulus conditions. But in general, the activity of single neurons tends to be heterogeneous and time varying. And it's not just the case in the prefrontal cortex. People have also looked at the gustatory cortex, which has been shown to play a role in the representation of taste. So in this paper, the authors recorded from the gustatory cortex in mice, and they sampled 4 different taste stimuli. And they're showing the average activity for these 4 stimuli for 4 neurons. And we can see that in general, the activity tends to be quite dynamic, even at a single neuron level, and if we consider the average. And moreover, in the gustatory cortex, people have also observed that there tends to be switching between states. And what I mean by that is that the population of neurons that they record from tend to change their firing rate simultaneously. And it's also demonstrated here. So in this state, the population of neurons tends to have this firing rate profile, and it changes across states. And the sequence of states might correspond to different neural processes, like the representation of taste, which has been further evidence that the representation might be changing over time. Now, as neuroscientists, when we try to answer these questions, we typically don't know how the brain is encoding information. And we have access to this data that's just the recording of neurons across time for various repetitions of a task. And we try to answer these questions, like what are the sensory features that drive this neural response? And what features of this neural response drive behavior? And one way that we can answer this question is by using decoding, which in more practical terms just corresponds to building something like classifiers, and then decoding features of the stimulus, like the position of the animal, or trying to decode the decision of the animal. And since, based on the observation that the activity tends to be time varying, people have commonly used time varying decoders. And I'll just go over what that means in a second. So let's take this paper in which the authors trained animals to match a sample stimulus to one of two categories. And once again, they had to maintain information about the sample for several seconds. And they recorded from two areas, including the prefrontal cortex. And then they tried to look at the time course of this encoding by trying to decode these two categories from the population activity that they recorded from. But the more important point here is that they build an independent decoder at each time bin to classify one of two categories, which implicitly assumes that this representation is changing over time. And this approach has also been used in other papers. For instance, in this case, where the authors tried to classify different taste stimulus using the population of neurons recorded from the gustatory cortex. And once again, they trained separate decoders at each time bin to look at the time course of classification and encoding. And what's the problem with time-varying decoders? Let's say we have two stimuli and they're being encoded using the firing rate. Now, the stimulus can be decoded by downstream areas in the brain if they know the timing in the trial. Which seems like it's not a very complex condition, but in fact, if we look at the neural circuitry, like let's say these are three neurons and the activity is changing with time, and they have synaptic connections with dendrite of some downstream neuron that's reading out the stimulus. Typically, the synaptic weights would be fixed. So if they have activity that's changing with time, these synapses would have to dynamically change, or it would require more complex circuitry. So it's not entirely biologically plausible. So what's the alternative? So we know that downstream areas are able to reliably decode information because the animals are able to perform behaviors. So in some way, the neural circuits are able to maintain this information at a population level. So how could this happen? And one possibility is that this could happen because there is redundancy in the neural population code. And by that, I mean that it's possible that the encoding does not span the entire space of neurons, but in fact, it's restricted to a smaller space. And I once again go over it some more in the next slides. So this particular idea was explored in this paper in a linear network. So let's say we have a stimulus and it's we can assume that it's being coded by the weighted sum of the activity of each neuron, which is given by A. And these are just the feature vectors for each individual neuron. And we want the activity to be constant in time. So we want the time derivative of S to be 0, and we get this condition. And this is how they model the dynamics of each individual neuron in this paper, where L is the connectivity matrix. So Lij would correspond to the connectivity between neurons I and J, and A and J would be the activity of the jth neuron. And if we just plug in the condition that we got on the previous slide into this, we get this equation. And when we just change it into the matrix form, and then assume that Vi and lambda I are the eigenvectors and eigenvalues of L, we finally get this condition, which is satisfied either if the eigenvalue corresponding to the ith direction is 0, which would imply persistent activity, or it could happen if Vi lies in the null space of D, in which case the activity can be time varying. And when they simulate from this network, they in fact see that some neurons are plotting the average activity for some conditions. Some neurons do show persistent activity, whereas some neurons have time varying activity. And this is just the broader idea of the paper where the activity in the coding space is persistent, and all the time varying activity is constricted to this orthogonal or null space. However, linear networks are kind of restrictive, and moreover, there is no stimulus information in the null space in this case, which would imply that if we train time-varying decoders or time-invariant decoders, they will have the same performance. And we'll see that this is not the case in the following slides. So here, the authors tried to extract a time-invariant stimulus space. And they train animals to perform this task where they had to maintain a target location for some delay, and then make a response to it at the end of the delay period. The only addition here was that there was a distractor between during the delay period. And then they tried to decode by training at different time points during the trial, and then looking at the performance for the entire duration of the trial. And we see that the performance is high along the diagonal, which kind of implies that it does not generalize well across the trial. So it's not picking up a time invariant code And it's also demonstrated here whether looking at the activity during the trial for 4 different target locations and projected onto the first 3 principal components. And we can see that the activity seems to be changing over time. And in order to find a time-invariant stimulus space, they solved this particular optimization problem where they wanted to find a subspace U that satisfies 2 conditions. One is that the activity between the 2 delay periods should be minimized. And it should also maximize the target information. And they do this by maximizing the projection of the conditional average condition on different target locations. So they try to maximize its projection onto you. And after doing that, they do see that in the subspace, the target locations are well separated. And moreover, the activities during the 2 delay periods tend to overlap. And if they try to do the cross-temporal decoding again, we can see that it's much more stable. Like if they train on a particular delay, it tends to do well for the entire duration of the trial. However, even in this case, this particular method requires identifying an invariant subspace before decoding. But it would be simpler to build a time-invariant decoder directly. Which brings me to the first part, where we try to build a time-invariant decoder. But before we can do that, we want to simulate some activity that satisfies certain conditions. And our broad idea is that we have a stimulus space where the activity is linear and it does not vary in time. So these are 2 example stimuli. And we have an orthogonal space where the activity is nonlinear and time varying, but it could still have some information about the stimulus, or it could be different based on the stimulus condition. So we construct 2 modules with 2D dynamics, and in both of them, the invariant signal is just defined as a standard normal. And we have different means for different classes. And in the first module, our orthogonal time-varying dimension is constructed using a sinusoid. And we use different phases to differentiate between different classes. So we assume that 1 second is where we have a stimulus onset, and there's a change in phase. And one reason for picking this phase is because previous studies have seen that if they disrupt the ongoing phase of oscillations, they tend to see a difference in behavior, which they measured using reaction time. In the second module, we constructed the orthogonal dimension from a renewal process with 5 states, where the inter-arrival time is sampled from a gamma, and this is the example states from 1 class. And this was done to kind of mimic the observations from this previous study that I talked about, where the authors observed switching of states. And this is what the second renewal process looked like for all classes. It's kind of messy. And we simulated our observations for 300 neurons using a Poisson process. And if we,
So I have a quick question.
yes.
So the sinusoids, they are phase shifted, same frequency, but amplitudes are also the same? I mean,
Yeah, yes.
I see. Okay.
Okay, so when we look at just the example neurons that we simulate from for both the cases, I'm just plotting 3 here. The activity is time varying both for the average response and for individual trials. So next, we got to the decoding part. And first, we're going to try to do a time varying decoder. So we have a baseline of comparison. And in order to do that, we first pre-process the observations. We can either do this by binning or reducing the dimensionality. And this is what our data would look like, where this is the entire trial duration of the stimulus onset. And we have some windows, depending on how we bind our data for different trials. And this would be either across the number of neurons or the number of latent dimensions in case we do any dimensionality reduction. And in the time varying case, we build a separate decoder for each window. We use a multinomial logistic regression in this case with L1. And we test it out on left-out trials. But as I said, it requires a separate decoder for each time bin. So after training, and we plot the performance on the test set, once again, this is like a cross-temporal decoding matrix. So this would be the time of training, and then I'm just plotting the test performance for the entire trial. And we can see that in both cases, it does well along the diagonal, but it doesn't generalize very well for the entire duration of the trial, which means that it's kind of overfitting to the window that we're training on, and it fails to find the time-invariant stimulus space, even though it does have access to it. Next, we went on to building a time-invariant decoder, which was exactly the same except for how it was trained, where we use samples from different time bins as independent samples to train the decoder. And then once again, tested it on the left-out trials. Sorry. And when we look at the performance for the single decoder, we see that it does quite well for the entire duration of the trial compared to the time varying case. So it's able to ignore the information in the null space and focus on the time invariant stimulus information. So the conclusion for the first part is that I propose a simple decoder that is able to extract a time invariant stimulus subspace directly. It requires less number of parameters. They're exactly scaled by the number of time bins. And from a biological point of view, this is good because the downstream neurons can decode stimulus information, even if they don't know the time in the trial. So next, we wanted to see how this time invariant decoder performs on real neural recordings. So to answer this question, we analyzed data that was collected from mice from the gustatory cortex while they sampled 5 tastings. And the activity was recorded using imaging, which gives an indirect measure of the spiking activity. And this is what it looks like. I'm just plotting it for 2 neurons. The number of neurons ranged from about 147 to 448, and we had about 50 to 60 trials. And
so I have another question. Sorry about that. So, are you sampling individual neurons? I mean, or a collection of them?
So in this case, we have recordings from the entire population.
I see. Not that you know exactly what every individual neuron does, right? You're just population level sampling. Is that so?
I mean, we assume that we have the activity from each of these individual neurons. They're just recorded simultaneously.
Oh, I see. From each of them.
Mm-hmm.
I see. Okay. Okay.
So we first reduced the dimensionality to kind of reduce the complexity of data using PCA or NMF. And then once again, we fit the time-varying decoder that I described previously. In this case, I use 1.2 second windows. And when we look at the performance, I'm plotting the decoding accuracy over time. And this is the decoding performance for 6 separate decoders. The dotted lines represent the midpoint of the bin that was used for training. And we can see that the decoding performance is high for the window that was used to train, but it does not generalize very well. So the performance decays quickly.
So yes.
Yeah.
So I mean, maybe I haven't entirely understood this. So here, when you are decoding within that time bin, right? You are trying to decode individual neurons. Is that what it is?
I'm trying to use the population of neurons to decode 1 of 5 different tastings.
Okay, so it's not exactly individual neurons. It's a population of neurons.
Yes,
So I'm getting confused over there, right? Okay, so in that case, does it mean that I'm looking at this collection of sinusoids, right? So you are trying to find the upper envelope of this? Is that what you're trying to find?
Oh, no. I think I just didn't explain it well, my bad. So in this task, there were 5 different stimuli, and we have recordings from, let's say, some N neurons. And these recordings are across some time.
Okay, over there are five. Okay, I see. Can you go back to that plot? Not that. The original one, the 5 signals. There are 5 of them. That's what you're saying.
No.
6 of them.
No, I'm just plotting 6 example decoders. So I'm training a different decoder for each time bin.
Yeah, so that So now the question is, each decoder is trying to do what exactly? So it is really sampling the sinusoids, isn't that so? Trying to decode the sinusoids, amplitude of the sinusoids, or the upper envelope?
Oh no, in this case there are no sinusoids. It's just trying to decode 1 of 5 stimuli, so 1 of 5 values. It's just a categorical classification.
I see.
So if it picks up on a common stimulus space, we would see that it classifies well for the entire duration of the trial, but it doesn't but it tends to overfit to this window.
Mhm.
So even though I'm training on this window, I'm just plotting the performance for the entire trial to see how
Okay.
well it generalizes across the trial.
Okay. Yes, I think I now somewhat understand it. Right, I was confused with the sounds. Okay, so you can go on.
Maybe this plot will be more familiar since I showed it before. So this is the cross-temporal decoding. The x-axis would be the window that was used for training, and the y-axis is the test, the entire duration of the testing trials. And we can see that both for both semi-NMF and all data, the decoders do well around the time they're trained, but the performance tends to decay quickly, or it doesn't do as well for the entire duration of the trial. These are two different sessions. And next we wanted to see if we can get a similar performance using a time-invariant decoder, which was trained in a similar way that I described previously, where we used each window as an independent sample to train the classifier. And if we look at the decoding performance, so the reason why there are these three separate sections is because I trained 3 decoders. So one of them was for this about 0 to 8 second interval, and then this was about 8 to 16, and this was before stimulus. And then I'm plotting the performance for the test trials. And we can see that this does tend to generalize better because it's able to pick up on a common stimulus space. And if I just plot the second decoder, we can see that the representation is stable for about 8 seconds for both sessions. Now just to compare it with one of the time-varying decoders. So the blue one is one time-varying decoder that was trained around this time. And we can see that it decays much quickly than the time-invariant case. And both of them were trained using all data, and there was no dimensionality reduction. And moreover, if we plot the average performance from 0 to 10 seconds for the time-varying and the time-invariant case, the performance for both of them is comparable for all 3 cases where we use dimensionality reduction, or if we include all data. And just to go a little further into how the time-invariant decoder is able to perform and generalize well across the trial. Here I'm just looking at 2 example neurons from each session that had very high decoding rates in the time-invariant case, and we see that the activity for both of them is not persistent, conditioned on the different tastings, but in fact it's variable in time, which suggests that the time-invariant decoder is not just using a single neuron, but it's relying on population code And we can also see this if we try to decode using that single neuron and compare it with the performance where we use all neurons. So it doesn't do as well if we just train it on the best neurons. Okay, so this brings me to the conclusion for part 2. We showed that time-varying decoders do not falsify the existence of a time-invariant neural code. We showed that a time-invariant decoder is able to decode stimulus information even when the underlying activity is time-varying, when it is maintained in a low-dimensional subspace. We also showed that in the gustatory cortex, the population activity indeed has time-invariant representation of taste that is stable over the course of seconds. But a more broader conclusion would be that just because we can decode, it does not imply that the downstream areas or neural circuits are using that information. And we would need further experiments to verify the coding scheme that is being used by neural circuits. And with that, I would like to thank my lab, specifically my advisor, Maming, for his guidance and support, and everyone else in my lab. And I'd also like to thank Dr. Fontanini, as well as Ko and Josh in his lab, who provided us with the data that I analyzed. And with this, I'd be happy to take any questions.
A question. So based on your findings and observations, would you conclude that the time varying and time invariant signals really do lie in a linear subspace, or do you believe that maybe they lie in a nonlinear manifold? Or
I think it's hard to say from this particular study if they lie in the linear or nonlinear subspace. I think it would depend on the area and the task that they perform. It does seem like, at least in the gustatory cortex, is a linear or time-invariant decoder is able to do well, which suggests that they might lie on linear space, but I think it's hard to generalize that just based on this.
fair enough. Fair enough.
So I've got a quick question, but just, you've got a model. What does this tell you about that part of the brain in general? I mean, what can we, I hope this is a well-posed question, but what in the big picture does this get you?
I mean, like I said, in the decoding case, we can say that the brain has access to this information. We don't know if it's using it in any way. We can just try to parse what signals the brain has access to. And the kind of questions we ask are different based on the different areas that we record from. Like in the gustatory cortex, we want to ask how is taste being represented, because people have shown that it plays a role in taste. But without perturbing the activity of neurons, it's hard to make any broad causal statements that this is what the brain is doing and this is how it's doing it. Especially with decoding, I think decoding is just like you can't make any strong claims with decoding analyses.
Okay, thanks.
Any other questions? Well, if not, let's thank Ayesha again, and thank you all for coming.
Thank you so much.